Hello, fellow warriors. Here we are with news from the website prostatewarriors.com. Uh, what if the reason we cannot seem to cure certain cancers isn't because our drugs are, too weak, but because the cancer simply knows how to play dead?
Right. That is the exact question researchers are grappling with.
Welcome to this custom deep dive. Our mission today is to take a massive stack of recent oncology updates, research papers, and trial data, and, well, distill it all down into actionable, understandable insights just for you.
And it really is a formidable stack of research to get through this time. I mean, the density of the science is high, but it points to a fundamental shift.
A shift in how we actually fight the disease.
Exactly. We're moving away from the old paradigm of simply blasting tumors with blunt force and finally beginning to decode their intricate metabolic and genetic vulnerabilities.
That shift is exactly what we are going to trace today. We are going to start right on the immediate front lines with the latest clinical trials, the therapies actively being tested right now.
Yes, and then from there, we will look at how we use genomics to personalize existing treatments.
Right. And finally, we will explore some truly mind-bending preclinical science that might hold the key to curing the previously uncurable. So let's start on those front lines. The first major update is the Phase 3 PRTK03 trial.
The PRTK03 trial.
Yeah. This one is aimed at localized prostate cancer, meaning, it hasn't spread beyond the prostate gland yet. Patients received a drug called Aglatimagene Besadenovec, which is often referred to as CAN-2409, thankfully.
Much easier to say.
Seriously. And that was combined with an antiviral medication called valacyclovir. This was all administered alongside standard radiotherapy.
Right. And the mechanism behind CAN-2409 is just a masterclass in biological manipulation. It is an intratumoral adenoviral gene therapy.
Okay, let's unpack this. What does that actually look like in practice?
Well, in practical terms, researchers take an adenovirus, strip out its ability to replicate and cause illness, and use it purely as a delivery vehicle.
Like a microscopic FedEx truck.
Exactly like that. They inject this modified virus directly into the tumor, and the virus inserts a specific gene, the HSV-TK gene, into the cancer cells.
Okay.
Once that gene is incorporated, the cancer cell starts manufacturing a completely new enzyme.
I mean, it is a Trojan horse. You use the virus to sneak a new set of instructions into the tumor. The tumor thinks it's just going about its normal business.
Right, it's completely unaware.
But then the patient takes valacyclovir. Now, valacyclovir is usually just a mild antiviral pill people take for cold sores.
Yes, a very standard benign medication.
But because the tumor has been reprogrammed by that viral payload, it reacts totally differently to the pill.
Precisely. The newly manufactured enzyme inside the cancer cell grabs that harmless valacyclovir and metabolizes it into a highly toxic nucleotide analog.
Wow.
It essentially builds a cancer-killing weapon inside its own walls, destroying its DNA from the inside out.
So it poisons itself.
It does. And when combined with standard radiotherapy, the results from this Phase 3 trial were remarkable. The hazard ratio was 0.70.
Let's sit with that number for a second, because a hazard ratio of 0.70 carries incredible weight for a patient.
Highly significant, yes.
That means a 30% lower risk of disease recurrence or death continuously over the entire follow-up period. A sustained 30% drop just by tricking the tumor into poisoning itself. But looking at these trial notes, I have to push back here.
Okay, go ahead.
I mean, I have to ask this passionately. This trial started 14 years ago. 14 years.
That's a staggering amount of time.
For someone fighting this disease today, that is a lifetime. How is it acceptable that patients have to wait over a decade for these advancements to reach standard care? I mean, how many recurrences could have been prevented if we had better early surrogate endpoints?
The frustration is entirely justified. It really is. And it points to a systemic flaw in how traditional oncology trials are designed for localized prostate cancer.
Because the disease moves so slowly.
Right. Prostate cancer often progresses very slowly. While it is wonderful that patients live a long time with localized disease, traditional trial designs demand median overall survival data.
Meaning you have to wait for people to literally die.
Yes. That means researchers literally have to wait until half of the trial participants pass away to statistically prove the drug extended life.
Yeah.
That takes over a decade. What the oncology community desperately needs are regulatory pathways that accept earlier surrogate endpoints, things like early PSA kinetics.
Meaning tracking the velocity of how fast a patient's PSA levels rise early on, rather than waiting a decade for the final grim data point.
Exactly. If the FDA and other regulatory bodies accepted advanced imaging metrics or the speed of PSA doubling as definitive proof of a drug's success, well, We could get that 30% risk reduction into the clinic years faster.
Which would just change everything for so many guys. If we are talking about systemic flaws and rethinking our approach, that brings us perfectly to our next trial. Let's look at what happens when the cancer has already spread.
Right, moving into metastasis.
Because for decades, oncology dogma has dictated that once metastasis occurs, you just ignore the prostate. The prevailing thought was that the horse has left the barn, so you just flood the body with systemic therapies.
But the phase 1 RIALTO trial completely upends that thinking.
It really does. How does RIALTO change the game?
So RIALTO challenges the old dogma by exploring what is known as the oligoresidual concept. This trial looks at patients with de novo metastasized prostate cancer, meaning the cancer had already spread to other parts of the body by the very first diagnosis.
Okay, right.
Specifically, it targets patients who show an incomplete response to hormone therapy, where their PSA levels refuse to drop below 0.2 nanograms per milliliter after 6 months of treatment.
So the therapy is working, but not quite finishing the job.
Exactly.
Yeah.
The hypothesis here is that the primary prostate tumor isn't just an empty original nest. It actually acts as a persistent tumor reservoir.
Oh, I see. If you have a leaky pipe flooding the house, you don't just mop the floor, right?
Yeah.
You have to fix the pipe itself.
That's a great way to put it, yes.
Yeah. Or like a dandelion in your yard. If you only focus on blowing away the white seeds that have scattered across the grass, you are ignoring the deep, thick taproot still buried in the soil.
Right. And to find every seed and map out the root, the RIALTO trial relies on highly sensitive PSMA PET-CT imaging.
Because PSMA is heavily expressed on the surface of most prostate cancer cells.
Exactly. By injecting a radioactive tracer that binds to PSMA, doctors can light up the cancer on a P-scan. This allows them to see sub-millimeter bone lesions and lymph node involvement that older traditional CT scans would completely miss.
It gives them a perfect map.
Yes, and with this high-resolution map, they precisely guide radiotherapy to the distant metastases and back to the original prostate tumors simultaneously.
But there is a well-known catch with PSMA PET scans, isn't there? They aren't flawless.
No, they are not.
And that brings us to the phase 1b to MVB-101 trial.
Right. The catch is biological heterogeneity. While most prostate cancer cells express PSMA, up to 20% of circulating tumor cells could be PSMA negative.
Meaning if they don't wear the uniform, they are essentially invisible to the PE scan and totally invisible to any drug that only hunts for PSMA. They just sit there, hiding in plain sight.
Exactly. And enter MVB-101, also known as CBP-118. It is a bivalent, peptide-like drug conjugate.
We need to break that term down because it sounds like a mouthful, but the engineering is actually fascinating. Bivalent means it has two different targets, right?
Correct. Dual targeting.
And peptide-like drug conjugate. Normally, we hear about antibody-drug conjugates, which use large immune proteins to deliver toxins. But MVB-101 is tiny, isn't it?
It is incredibly small, about 1/50th the size of a traditional antibody-drug conjugate.
Wow, 1/50th. Why does that matter?
Because solid tumors are dense, highly pressurized masses of tissue. Large antibodies often struggle to penetrate deep into the core of the tumor. Because MVB-101 is built from small peptides, it can diffuse deeply and rapidly right into the tumor tissue.
Okay, that makes sense. And what is it targeting?
It looks for PSMA, but crucially, its second target is folate receptor alpha, or FR alpha. When it finds a cell expressing either of these, it delivers its payload, which is a tubulin inhibitor called MMAE.
And tubulin is basically the internal scaffolding of the cell. If you inhibit it, the cell cannot physically divide and replicate. It paralyzes the cancer.
Yes, it completely stops the division process.
But the choice to target folate receptor alpha as the backup mechanism, that is the genius part to me. Why target folate?
The biological rationale is incredibly elegant. Cancer cells are defined by rapid, uncontrolled division. To divide, they have to synthesize massive amounts of DNA, and folate is an absolute requirement for DNA synthesis.
Oh, I see.
The folate receptor is hardwired into the cancer cell's core metabolism. If a cancer cell mutates to drop its PSMA expression, it can hide from standard drugs relatively easily.
Right.
But if it tries to drop its folate receptors to hide from MVB-101, it essentially starves itself of the nutrients it needs to survive.
It is a metabolic trap. You can take off your camouflage, but you can't take off your lungs.
Precisely. It corners the cancer.
Now, we've talked about delivering toxic payloads and using viral Trojan horses. Let's take a leap into the ultimate frontier of precision medicine, which is editing the genetic instructions directly.
This is truly science fiction becoming reality.
It really is. And this brings us to a phase 3 trial. Now, This is not a prostate cancer trial, listener, but
Yeah.
the implications for the entire field of oncology are staggering.
The trial in question is for a rare, life-threatening genetic disorder called hereditary angioedema.
Right.
It causes severe, unpredictable swelling attacks. The trial used a CRISPR drug called lonvigurin-ziclumeran. Instead of extracting cells, editing them in a lab, and putting them back in, they infused the CRISPR therapy intravenously directly into the patients.
Inside the living body.
Yes. The drug traveled to the liver, found the specific kallikrein gene responsible for the disorder, and permanently switched it off.
And it reduced swelling attacks by 87%, editing the genome of a living human being in real time. So, what does this all mean for our listener fighting prostate cancer? Why do we care about a swelling disorder?
Because the liver is the body's primary manufacturing hub. It produces a vast array of circulating proteins, hormones, and metabolic factors that travel through the bloodstream.
Okay, so it affects everything.
Right. For years, the barrier to CRISPR was proving that in vivo delivery editing inside the body was safe, targeted, and effective on a large scale. By successfully executing this in a phase 3 trial, they prove the platform works.
Which changes the game entirely.
It opens the door to treating a massive range of genetic and metabolic diseases. Eventually, we could use in vivo CRISPR to directly target the systemic, genetic mechanisms that allow cancer to thrive in the body.
It is awe-inspiring to think about. But editing genes is the future. Right now, the most immediate power we have is reading them.
Yes, reading the genetic signatures we already have.
Right, because if a doctor could read a tumor's exact genetic signature today, how would that change a patient's treatment plan tomorrow? That brings us to the Decipher Genomic Classifier.
Decipher is a very powerful tool. It is a 22-gene RNA assay.
Okay, how does that work?
To understand it, think of DNA as the master blueprint locked in the cell's nucleus, and RNA as the individual work orders being sent out to the factory floor.
Oh, that's a great way to look at it.
By analyzing the RNA expression of 22 specific genes in a tissue sample from the primary tumor, the Decipher test sees exactly what the cancer is actively building. It outputs a score based on that activity.
And researchers have been using the score with trial data recently, right?
Yes, they looked at retrospective data from the ENZAMET trial, which studied men with metastatic hormone-sensitive prostate cancer. They found that patients with a Decipher score above 0.85 see a massive survival benefit from triplet therapy.
Now, triple therapy is an aggressive combination of androgen deprivation therapy, a secondary hormone drug called enzalutamide, and a harsh chemotherapy drug called docetaxel.
Right, it's hitting the cancer with everything we've got.
But here's where it gets really interesting. Historically, these genomic tests were viewed mostly as prognostic tools, right?
Yes, just guessing how bad the cancer is.
Right. A doctor would look at a high score and say, this cancer is highly aggressive, things look tough. It was just a way of guessing the future trajectory. But this ENZAMET data flips the script. It shifts the Decipher test from purely prognostic to treatment selection.
That distinction is vital. Chemotherapy is a systemic sledgehammer. It carries severe toxicities that impact a patient's entire quality of life.
Absolutely.
The data suggests that if a patient's Decipher score is below that 0.85 threshold, they might safely skip the docetaxel and stick to the doublet hormone therapy without sacrificing survival odds.
We are finally using hard, objective data to spare patients unnecessary suffering. We are treating the tumor-specific genetic signature, not just defaulting to the maximum tolerable dose of poison.
Exactly. It's a much smarter approach.
But even when a patient gets that perfect genome-guided triplet therapy, even when the scans come back totally clean, we still see tumors relapse. Why?
It is the most frustrating hurdle in oncology.
Where does the cancer come from if it was completely gone?
The answer lies in the last 0.1% of cancer cells, the Persister cells. And this brings us to the absolute bleeding edge of preclinical science. Researchers at UCSF recently developed a high-throughput mini-tumor platform to study these extremely rare cells.
Microscopic tumors in a lab?
Yes. They grew thousands of microscopic prostate tumors, subjected them to standard therapies, and isolated the tiny fraction of cells that survived. Crucially, their findings force us to rethink the mechanics of drug resistance.
Because normally we picture drug resistance like, Darwinian evolution. You
Wow.
apply a drug, it kills 99% of the cells, but one cell happens to have a random permanent genetic mutation that makes it immune.
Right, the classic mutation model.
That mutated cell multiplies, and suddenly you have a resistant tumor. But UCSF found something different.
Completely different. The UCSF study revealed that Persister cells don't rely on permanent genetic mutations. Instead, they enter transient, temporary survival states.
Wait, temporary?
Yes,
Yeah.
they alter their epigenetics and their metabolism to essentially shut down and weather the storm.
Oh, wow. So it's not a criminal permanently mutating into Superman. It is like a submarine under attack.
A submarine.
Yeah. The submarine turns off its engines, kills the lights, drops to the dark ocean floor, and just runs on low-power emergency batteries until the destroyers pass overhead. They just hunker down.
A perfect analogy, yes.
Mhm.
But the UCSF researchers did not just observe this behavior. They tested those surviving, hibernating mini-tumors against 94 different chemical compounds.
And did they find anything that works?
They did. Because these resistor cells all rely on similar transient metabolic states to survive, they share common vulnerabilities. Out of the 94 compounds tested, 9 successfully targeted and killed the persistor cells.
They found the depth charges that work while the submarine is hiding. If we know what the vest is made of, or what metabolic state they are using, we can pierce it while the tumor burden is at its lowest.
Exactly. Right after standard therapy finishes. The challenge, however, is getting the body's immune system to help with that strike.
Ah, right. Because prostate cancer is notoriously an immunologically cold tumor.
Yes, meaning the immune system's T cells essentially walk right past the prostate tumor, completely ignoring these hiding persistor cells.
So how do we blow their cover? How do we take a cold tumor and force it to turn hot?
We turn to some brilliant preclinical work out of Cornell University involving silica particles, or CDOTs.
Prime dots, what makes them so special?
Well, these are ultra-small silica nanoparticles. The beauty of these silica particles is that they have already been proven safe in humans. They were originally developed as kidney-filtered imaging agents.
So the safety profile is already there.
Right, but the Cornell researchers engineered them to become weapons. They coated these tiny silica dots with molecules that target PSMA.
Okay, locking onto the cancer.
And as the C dots are absorbed into the prostate cancer cell, they drag a massive amount of iron ions in with them.
And a sudden influx of iron inside a cancer cell. That triggers a very specific chain reaction, doesn't it?
It does. It triggers ferroptosis. Now, normal programmed cell death is called apoptosis, which is neat and quiet. The cell shrinks and is recycled.
Right.
Ferroptosis is a violent, chaotic cell death driven by lipid oxidative damage. The influx of iron essentially causes the cell to violently rust and burst open.
And when a cell bursts via ferroptosis, it releases those oxidized lipids that activate toll-like receptors, alerting the immune system.
Exactly. The immune system is jolted awake by the massive tissue damage.
So we are basically sneaking a biochemical flare gun into the tumor, forcing it to explode, and finally waking up the immune system to do its job.
Yes. And when combined with 2 checkpoint inhibitors and a CSF1R blocker, this method achieved about a 50% complete remission rate in aggressive mouse models.
A 50% complete remission rate just by pulling the fire alarm. That is incredible.
It truly is.
Well, let's take a breath and recap the incredible journey of this deep dive. We started with the slow but triumphant 14-year Phase 3 PRTK03 trial, rethought how to treat metastases with RIALTO and MVB-101, explored the power of the Decipher test, and finally looked at how forcing ferroptosis and targeting persister cells could change the future of the disease.
The sheer volume of data is immense, but the core takeaway here is empowerment. Knowledge is a weapon. Understanding the why behind these treatments helps you make much better decisions with your medical team.
Absolutely.
And I want to leave you with a final provocative thought today. If cancer cells can enter transient metabolic states to hide from our best drugs, and we can trigger ferroptosis to violently blow their cover, where does it end? Could the ultimate cure someday involve mapping the metabolic states of our own healthy cells to prevent cancer from ever taking root in the first place?
Master the environment so the seed never grows. That is a profound vision of the future. Thank you all so much for joining us on this deep dive. Stay strong and fight on.